NGS workflow. And the way I have structured the presentation is initially to talk to you briefly on what the sample requirements are like for Illumina platforms. So if you're going to be doing NGS on Illumina platforms, there are certain basic standards that you need to meet. And should you have doubts or find out that you don't meet those standards, we have certain suggestions for you to clean up your samples, and then also for quantitative and qualitative determination of your nucleic acids. We have some solutions. And finally, I will talk to you a little briefly on library preparation as well as quality control for your libraries. Can I just ask everyone to mute yourselves, please, just so that we can proceed? Right, thank you very much. Okay, so for some of us who are hands-on on next generation sequencing, and those who are still learning about it, I'm sure you are, as we go along this webinar series, discovering how intensive it is costly, either in cost of kits and all the necessary consumables and equipment that go with it, but also very costly in time. And the entire goal of the workflow is so that you end up with DNA fragments, double-stranded DNA fragments of a specific length and purity, as well as concentration that are compatible with our sequencing platforms of choice. In this case, we're talking about the Illumina platforms. And so quality control is very essential in making sure that only those particles or fragments of the suitable quality proceed and get sequenced, so that eventually when you do get your library, which is very key in the success of your sequencing runs, you're making sure that you're getting there with just the best of your sample, and that your library quality is very high and premium to guarantee that you get the sequence reads that you want, and also that they are valid to give you the kind of insight that you want from your research. And so the workflow usually proceeds beginning with, just want to take the pointer, beginning with your starting material and then ending up with your library. And the quality control steps will mainly be to control the quality of your samples in terms of quantity, that's the concentration, the purity, as well as their integrity. But also, once you've prepared your library, you want to guarantee that or ensure that they are of the right fragment sizes and within the right concentration, so that when you amplify, you get everything ready for your cluster generation. Just also, once you've amplified, there's another final step of cleaning up, taking away all your unligated dimers, adapters, or primer dimers, as well as just making sure that your fragments are within the right size required. So in this section, I just want to give you some checklist points of what is usually required, especially if you're sending your samples off to core facilities for sequencing, but also if you are doing the sequencing yourself, just those little checklists that you need to just keep in mind as you prepare your samples to send for sequencing. So like I said, it's a checklist. I'm not going to go into the details point by point. When you receive the slides, you can go through them and build up from there. But just to highlight that you might want to look into how you store your samples and handle them prior to submitting them for sequencing, because that may affect the quality of your sequence libraries. You also want to make sure that they are free of contaminants and within the right concentration and purity, as well as being submitted in the right type of buffer. And pH of that buffer is also very important to maintain the integrity of your samples. Right, so if you are interested in a specific type of library, there are different variabilities in that general recommendation. So this table kind of takes you through that. For your RNA samples, for those who are doing RNA sequencing, there's also some recommendations as well in terms of your purity, as well as your concentration, pH, and the recommended buffers for submission. Just to highlight here as well that for RNA samples especially, sometimes there is different workflows for isolating RNA. It's not always optimal to use the general or the most recommended spin columns, just because yield is an issue and also integrity is very key to RNA sequencing. So if you are going to proceed with organic purification of your RNA, then there might need to be an extra cleanup step just to make sure that you get rid of those potential enzyme inhibitors. Right, so for the different types of RNA libraries, you also have different recommendations, both in the integrity as well as quantities that are recommended. I'm not going to go into all of this. When Maria and the team take you on to next week, the next 2 weeks, looking at the specific platforms and the applications, as well as the library preparations for those applications, we're going to go into more details into that. So in certain cases, you may want to actually submit pre-constructed libraries. I know most people shy away from this, but it might be cost effective to actually buy your own library prep kits, prepare your libraries, and then send them off. So when we talk about preparing library, and we'll discuss that in a little bit more detail later on in this presentation, you want to make sure that they are of the right quantity, within the right size for your fragments, and that they're also free of some of the contaminants that are usually introduced during library preparation. Concentration is also key here, as well as the right buffer type for submitting your samples. And in certain cases, you may also want to submit or you want to do sequencing using some customized primers. So there's also recommendations there for that. So I put these long lists just for you to use as a guideline as you proceed with your research. It might be handy. It came from websites for certain core facilities that do sequencing for people. So I just thought it might be handy to have that as a checklist every time you want to submit your samples. Okay, so before we proceed into the actual quality control, certain times last week we saw how to isolate samples from different sources, but it's not always ideal, or you don't always get to use the best quality of extraction procedures for one reason or another, and there might be a need to clean up your samples a little bit further. Why you want to do this is because your samples usually come with built-in inhibitors for your PCR reactions and other enzymatic reactions that are very much at the core of sequencing or next generation sequencing. And so you want to get rid of your polysaccharides, your fats, your humic acids from soil, your heme from blood. So if you are not, if your sample isn't optimally purified, there might be a need for an extra cleanup step. Another reason why you may need an extra cleanup step is that certain treatment or extraction methods in themselves introduce certain potential inhibitors into your samples. and you want to look out for them because they would cause all of these sort of inhibitory effects. Sometimes they'll cause enzyme denaturation, or they may degrade your template, or may just compete with your template or with your cofactors. So you want to get rid of those. So Macquarie-Nagel, and you might be familiar with the name by now, offers you certain cleanup kits. So in the first instance, if you're looking to clean up large size DNA from organic methods, enzymatic reactions, or from buffer exchange that are abundant with salts, phenols, and exogenous nucleic material, you will need the Nucleospin genomic DNA cleanup kits. Now, these are very effective in cleaning up your kits, getting rid of all of these, okay, cleaning up your samples, getting rid of all of these potential inhibitors, and also just ensuring high recovery of a wide range of samples. So your fragment sizes from 100 base pairs to 50,000 base pairs will be recovered very significantly. And also it has the option of an extra small column just for those really minute DNA fragments that especially come when you are dealing with, say, clinical or forensic samples. So it comes in quite handy. However, this kit is not the right one if you're looking to clean up your PCR mixes or gel fragments. This is an application data showing how samples that were spiked with phenylchloroform containing reagents at 1% were eventually purified, and we had different sized samples from 2,000 base pairs right up to 23,000 base pairs. And you can see here that the samples were pretty much very close to the perfect the sample, the original input. You can see the yield was very much comparable. The purity was within the right range of recommended purity for DNA samples going forward for NGS, as well as the recovery of those particles, even the very large ones. So the kit is quite effective in its range and in its recovery and the purity that it provides you. So the next option for cleaning samples would then be for your gel fragments that you've run a gel of your DNA and you want to recover that, or your PCR products if you're doing, say, amplicon sequencing. You want to clean up from all of the other stuff that is in your reaction mix. This kit will allow you to do both just with one binding buffer, and it is also very suitable in that it's not sensitive to all those PCR reaction additives. You get up to 80% and above recovery, even of very small fragments, and there's complete removal of your primers, and you end up with a very small elution volume, which means that your DNA at the end of this process is concentrated and you don't need an extra concentration step to proceed. So this is how it works. There is a binding buffer and there's options as well. Then you would allow your DNA to bind onto the column and you wash away all the contaminants, and eventually with an elution buffer, you would recuperate your DNA. So you see, it's a very simple three-step system. However, for large fragments, you might need to do several elution steps just to recover everything. And what is also interesting here is that with your binding buffer, you get a pH indicator because your small DNA fragments will bind preferably within this range, 5 to 6 of your pH. And so there is a color indicator on the binding, or when you're using the binding buffer, that will indicate that you're within the right pH range. So yellow would indicate that you're within the right pH range for optimal function of the kit. However, if you start seeing green and blue, then you're off a little bit in the pH, and you may need to regulate that by adding buffers or hydrochloric acid just to bring down the pH again and then proceed. So this is again just application of this kit. In the left-hand side, we're seeing here a gel of different fragment types that were within your PCR fragments, and they were spiked with primers. And then you can see in lane 1 on each of these categories is the unpurified original sample that was spiked with your primers, and you can see the primers here. And lane 2 and 3 in each case is where we've used the nucleospin gel PCR cleanup kit to clean up the sample. So the first category is for 645 base pair, 252 base pair fragment, and then even your 600 and your 65 base pair fragment. There's complete removal of your primers. In the right hand, we're just showing here from DNA fragments that were purified from 1% agarose gel using this same kit, and you can see that there's significant recovery of varied sizes of DNA fragments. Again, there's complete removal of primers and high recovery for a broad range of fragments. Right, so now we're going into the heart of how you actually proceed to do quality control of your nucleic acids. Generally, they are classified into 3 methods. So you have your optical methods, and you have electrophoresis as well as qPCR based methods with options within each category. The 2 optical methods that are used for quality control of your nucleic acid samples will be based on absorbance and based on fluorescence. Your absorbance method, it works on the De Beer. Most of you are familiar with this, but just to review, is that there's a De Beer formula that calculates the concentration based on the attenuation of incident light by the sample. Whereas with your fluorescence, it is light that is incident onto your sample, excites the sample, and then there's an excited or emitted light following excitation of the samples or fluorescent particles in the samples. And this emitted light is detected by a detector upon passing through a certain filter. And this will then, usually there is a standard curve that is plotted for known concentrations of nucleic acids, and then your sample concentration will be inferred based on that standard curve. Okay, so what are some of the advantages and disadvantages of each method? Here we can see that with the absorbance method, it's pretty simple. It gives you an idea of the purity of your sample. You can get right away the concentration, and it's also using very commonly available laboratory equipment. On the other hand, it doesn't distinguish between the different nucleic acids, namely DNA and RNA. It is not very sensitive. So if you're looking to detect or to quantify very little amounts of nucleic acid, then the absorbance will not be the best. Usually also in the presence of contaminants that tend to absorb within the same wavelength as your DNA, there might be an overestimation of the concentration. There's also usually not any information about the integrity of your sample. However, if you're looking to do absorbance, and we saw in the first introductory slide that actually it is a method that is recommended by Lumina for purity assessment of your samples, we can offer you the quick drop from molecular devices. It is a very portable system with a touch screen, and it has 2 sample ports. So you can either use a droplet, and that droplet can be right as small as 0.5 microliters, which is 500 nanoliters, or you can use a cuvette port for larger volumes. And the limit of detection can go down to 1 nanogram per microliter. The touchscreen is very user-friendly, and you can work in up to 6 languages. The read time is about 4 seconds from the time you put in your sample to getting a read, and the read will offer you information on the purity as well as just the concentration. So it does all the calculations for you, and you get the concentration value right away. So with the fluorescence method, it is, like we've said, very, very sensitive. You can go down to picogram per mil. It's also selective because most of the dyes, fluorescent dyes, are specific for double-stranded DNA, which makes it a very specific method. And it's also quite accurate for that reason, because it will specifically pick out just the DNA, double-stranded DNA, and give you the measurement. However, it's costly and it's time-consuming, and you also don't get information on purity or integrity of your sample, and your fluorescent dyes are usually quite hazardous to work with. So those are just some of the downsides of using the fluorescence methods. But again, it's the recommended method if you're going to be doing quantitation. You can get away with doing absorbance measurements for your samples in terms of quantitative measurements. However, you will then need to supply more than the required amount of samples if you're sending away your samples. So you have to overestimate or overcompensate for the quantitative data that you may get with your absorbance methods, just because it's not that sensitive and specific. So in our fort, as I'm sure you now know, we have a lot of things that we can support you with across board. So this webinar series was really just to show you how we as operations can accompany you throughout the journey, from the time you're getting your samples, you're analyzing your samples for quality control, and right up to doing the actual sequencing and data analysis, so we can take you throughout the journey. And so if you're going to do fluorescence measurements, we have from molecular devices, we have microplate readers that have fluorescence detection modes. These are the options here. And they come pre-configured with a protocol for your quantity picogreen assay. So the software is already pre-configured to run that assay. And you can basically detect right down to 100 picograms per mil of double-stranded DNA. So it's quite sensitive. And another advantage of this devices or these instruments is that they are not filter based. So they work with dual monochromators, and that allows you to fine-tune your wavelength selection for optimal output in your data. Right. So we're moving now on to the second category of methods for quality control. And those are electrophoresis-based methods, the first of which is using gels. So these would usually be polymer gels on a horizontal slab, and your DNA is applied in wells on the precast gel, and then on the electric field, your DNA would migrate, giving you bands that differentiate in terms of the size of the DNA fragments. And you would then proceed to staining that DNA with intercalating dye, and with that you can infer, based on size markers, you can infer the size of your DNA, as well as based on the intensity, you can kind of get information on the quantity of your DNA. So it is the most common analysis method for this category. It's low cost and it's quite simple just to check the quality of your sample, and also it will give you information on the size and the quantity based on the size markers. However, it's quite tedious and involves a lot of hands-on. It can be quantitative, but the quantitative data is usually not reliable, so you have to proceed again and do either absorbance or florimetric measurements of your sample to confirm the concentrations. Another thing is that it doesn't really inform you on the concentration of contaminants, and it consumes your sample, so you have to you can't be frigid with your samples here, or frugal. So within this category, what separations can offer you from Takara is the MOPIT 1 or MUPIT 1 electrophoresis system. It is a small compatible system with a power pack that fits here. At this point, they kind of join together, and it fits into your gel tank, and it's compatible with multi pipettes. It can allow you to run up to 104 samples simultaneously. It allows you to select over 7 voltages, and that is very interesting or useful in that it allows flexibility in terms of the length of your electrophoresis. It's a heat resistant material, so you can pour very hot gel onto this, and you also don't have to worry too much about overheating, and you can clean up with boiling water when you're done. It's also safe in that it doesn't start working if the lid is not on. And you can see here, this is the gel. It comes with a gel casting tray in which you can either do 1 big gel or 2, or you can put in all 4 combs. And I don't know how visible you can see from there, but there's different teeth sizes on your combs that allows for as many samples as you feel comfortable to run. It also has intuitive control. There's buttons here that you can check or press in order to control the time. You can pause in between runs, and you can also select your voltage. So still, for your electrophoresis, you also have the option of using a safer nucleic acid stain, which is the PronaSafe from Conda Labs. It has improved sensitivity over ethidium bromide. So you can see here at lower DNA concentrations, you get still better signal with the Conda PronaSafe. It's safer. Okay, and we have options as well for loading dyes from Solus Biodyne. You have the option of using double colored stains, either same color or different colors. They migrate with different fragment sizes of your DNA in the gels, as well as DNA ladders, either of 1 kilobase pair or 100 base pair, depending on what you're trying to do. And from Takara, we also have intermediate sizes of ladders. Okay, so the next category of electrophoresis methods is the capillary electrophoresis method. This is the method that is applied or used in the Agilent bioanalyzer. And basically how it works is that you have a chip. It works based on microfluidic technology, a chip with channels that are filled with sieving polymer and a fluorescent dye. And again, under electric field, the DNA fragments are selected or separated based on size. However, they move with your fluorescent intercalating dye, and once they fluoresce, the signal is captured onto a computer, and data analysis is intuitive or integrated, and you get an electrophoreogram of your sample. Okay, so the advantages of this method compared to other methods is that it's more accurate and reproducible. So if you compare it with the process of the gel electrophoresis, where you have to pour your gel, run the samples in the gel, destain the gel, read it in a scanning device or photograph it, and then get your data. This one basically does everything for you all in one, and it makes it more accurate therefore because it's more automated. There's also different DNA kits, so you can really expand on the fragment sizes. It has higher sensitivity, requires very little amount of your sample if you compare it to the gel electrophoresis. It's also very much faster, and you get higher resolution of your band. So because it's so small, it's on the chips, there is better cooling of your system, which prevents band spreading. That is usually a result of heating that happens during the electrophoresis. Obviously, in the electric field, there's heat that is dissipated. So if it's not well eliminated, it causes expanding of your bands, and you don't get such good resolution. So here you get very much higher resolution in your separation. However, on the downside, you cannot really use intact genomic DNA because it's limited on what fragment sizes can go into the chips. You can also not recover your DNA or RNA. It doesn't give you information on the purity of your sample, and it cannot really detect contaminants. And again, it's quite costly in terms of the consumables. Once you buy a chip, you have to use the chip. If you don't use all the wells in the chip, too bad. It's a once off. So you want to run all your samples or one sample, you're still buying the same amount of consumables, which makes it can be quite costly. In the third category of quality control for your samples is the quantitative or real-time PCR. I'm sure most of us are familiar with this. I'm just gonna bring this here for the sake of those who are still new in this field. So it's based on fluorescence technology, where you are integrating a fluorometer together with your thermocycler to detect fluorescence signal of an added fluorescent marker into your sample. And as your sample is being amplified by the thermocycling, you're getting amplification of the signal, and that gives you an idea of the quantity of DNA in the starting material. Right, so you have two options of your markers. It can either be probes. The probes are usually specific in that they are homologous to a certain sequence on your target gene, and that offers you higher specificity and reproducibility of your data. Both methods are quite sensitive in that because it's mainly based on amplifying your DNA. You can start with really small amounts of DNA and still get enough signal to infer back and measure the quantity of the original sample. So this is a very sensitive method of determining the quantity of DNA. The probe-based method is compatible with multiplexing, meaning that you can detect several targets at the same time or in the same sample. Sorry. The dye-based method is rapid and simple. It's more cost-effective, and it offers you information on each cycle as well as melting temperatures. The disadvantage on the probe method, again, it can be costly, and you need in both cases to do quite a bit of optimizing of your sample. So we have a few qPCR kits that we can offer from Takara, both for probe-based and for dye-based qPCR. I'm not going to go into the details. When you get the slides, you can reach out to us and we can tell you more about these options. So just to summarize on the quality control of your samples, we've looked at absorbance-based, fluorescence-based methods. Here will be your capillary electrophoresis, and the example here is the bioanalyzer. That's the most popular one. And then your gel electrophoresis and qPCR. So the different methods will offer you different types of information, and you must just choose based also on your budget and what of these parameters is most important to you. Right. So we are going to go now into NGS library quality control. Sorry, I'm a bit cold in here. So for your NGS library, the details of the method and the applications and the different options of kits for different applications to use on different sequencing platforms will be dealt with in detail by Maria and her team in the next couple of Fridays. But just as a refresher from Maria's first session is that when we talk about library preparation, basically we're talking about preparing our inserts or DNA fragments, either coming from DNA or from RNA following reverse transcription. And we want to end up with adapters at both ends so that they are compatible to the sequencing platform or sequencing flow cells. And those adapters will involve regions that include your primer binding sites or your indexes, which will distinguish between your different samples within the pool of samples or library. And then you also have the oligo sequences, or the sequences that will be binding to the oligo sequences on your platform or on your flow cell, sorry. So this part would then be what we refer to as the adapters. Right, so for library quality control, it usually comes down to two things. You want to make sure that you have the right size of fragments, because that's very critical for the sequencing process and cluster generation. Also the concentration, and usually the size will be determined by capillary electrophoresis, whereas concentration is usually recommended to use the qPCR quantification, unlike this for certain cases. You end up usually with your concentration in absolute values or nanograms per microliters, and you need to convert that to molar values. So this formula here will combine your concentration in those absolute values and your size to give you a concentration in molar values. So if you are going to be looking at size distribution measurements, you want to focus on having an electrophoreogram that has a single peak in the perfect situation. You have a single peak within the size that you are targeting. However, you may find that you also have certain other peaks, such as primer dimers, adapter dimers, or you may have higher molecular weight peaks. The primer dimers are not usually of concern unless they are quite abundant, then you want to get rid of them. The adapter dimers can be a problem because remember they contain the sequences that would eventually bind onto the oligo sequences on the flow cells, and so if they are available or abundant in your sample, they usually get preferentially amplified, and that will kind of mess up with your sequence reads. Also, you don't want to worry too much about your border bands, but you can definitely get rid of them by cleanup steps. For concentration analysis, usually, especially if you're going to be doing 16S library pools or PCR-free DNA sequencing, it's recommended that you do qPCR quantification using the Kappa Biosystems CyberFast library quantification kit for Illumina sequencing platforms. That's a mouthful. That kit basically is very specific in that it contains primer premix that targets your P5 and P7 flow cell oligo sequences. So it makes sure that only those library fragments that have been incorporated the adapters with these sections or sequences uh, get picked up and detected, and that's how you quantify. So it includes 6 standards, which is usually a tenfold dilution series of a linear 452 base per template, and you quantify these based on the principles of qPCR that we saw before, and then you get an average CQ score for each of the DNA standards, which is then plotted against the concentration log concentration to generate a standard curve, and then that standard curve will then be used to determine the concentration of your library. Right, so those are the methods for quality control or quantitating your libraries. But again, you wanna, if you see that there's some blips and blobs in your sample, you wanna clean it up. Generally, most of your library prep cleanup, most of your library preparation kits will recommend the ampere bits for cleaning up your samples and for size selection of your fragments. I'll just want you to allow me to introduce to you the McCurry-Nagel beats, which work in exactly the same manner, and you don't need a change in your protocol or existing hardware. And this is just data to show that whether you're looking at the concentration of the sample by various methods, you find that the beats work quite efficiently in the same manner as the ampere bit. You can see the figures are very closely tied together. And also the reads, and for varied GC content and insert sizes, again, very, very close and similar to your ampere bits. This is just a workflow description of how you actually proceed to do your NGS library preparation in general, and these are the different steps in which the NucleoMag NGS cleanup and size select kit from McCurry-Nagel would allow you either to clean up or to size select your libraries. So there are specific protocols. I'm not going to go into the details of it, but basically it works on varying the ratio of your beads to your samples to allow for the appropriate size selection. It also has protocols for allowing removal of adapter diamonds, which we've said is usually of concern. And you can see here how before cleanup and after cleanup, you definitely get rid of those adapter diamonds using the Nuclomag NGS cleanup and size select kit. You can also perform double size selection with a specific workflow or protocol. Again, just data showing you how this looks and how this is actually successfully done using the NGS cleanup kit from Macarinago. So this kit is highly efficient in recovery, even of very little amounts of sample. You can tune your size selection. It's convenient. You can use it for manual or automated processing, and you can work completely at room temperature during your cleaning up of the libraries. It's stable at room temperature. Okay, so I'll just round up now by recalling that if you are looking to do rapid assessment of concentration and purity of your samples, you can do absorbance spectroscopy. However, if you have very little amounts of sample, your fluorescent quantification will be the way to go. To assess the integrity of your samples, gel electrophoresis is recommended. For fragment analysis, such as for your libraries, what is very critical will be capillary electrophoresis. And then if you're looking at functional quantification, or you really struggle with very little input samples, then you can do qPCR. I hope that this has been helpful to you guys. And that was my story for today. You can, let me just share my screen. You can unmute yourselves. I think we have a few minutes to take questions, if there are any.
There was a question from Mrs. Zipfel. How much does one have to overestimate the concentration if using the absorbance method alone? Do you have a general guideline, 10 times, or is it simple and or instrument specific? I don't know if you have an answer to that.
So what I saw, usually what was recommended is that you just double the concentration. So if you have enough samples to be on the safe side, you double the concentration of what is required. That is, if you're doing your absorbance measurements, what you get, you double that up to make up for what is required. So I don't know if that makes sense. So if they say 10 micrograms, send us 10 micrograms. You measured 10 micrograms with absorbance, send 20, just to be safe, because there's overestimation of your DNA when you do absorbance measurements. Any other questions? Okay, if there's no other questions, I would like to thank you again for making time. I hope that you've signed up the participation, you've followed the certificate of participation link so that we can have you registered for this session. And we will be sending the slides. I'll share the slides with you in the coming days, and as well as a link for the recording. But also I'll send you more information on some of the products and instruments that I mentioned during this. But if you have any other questions that come up, please don't hesitate to contact us.
There's another question in the chat box. If you maybe want to Have a
Is
look.
that Maria? What are the sharing methods for DNA fragmentation into required size? Okay, so that is more into how you actually prepare your library. I'll just highlight that you can do that by sonication, or you can use the tagmentation, but I won't go into the details because Maria and the team, when they look at the details of library preparation and actually the sequencing protocols, will take you through that. Any other questions? So I think by the end of today, you would receive a link for registering for the next session. Next week, we're looking at the ISIC in the first session and then the mini-SIC in the next section. So it's going to basically be taking you through what the instrument does, how it works, what are the applications that can be done on the instruments, and then what kinds of questions you can answer there, and what are the specific requirements for that specific application and the library prep and the entire workflow. So I hope that you're as excited as I am to join Maria and the team on that session. Otherwise, have yourself a great Friday afternoon further, and an excellent and restful weekend. Talk to you soon. Bye.